Hi, everybody. Welcome back and welcome to the seventh and final session of this two-day workshop. So this session 7, the title is Growth, Development, and Aging. And I think this is going to be a really exciting one because it's probably going to bring together a fair number of the themes that we've been hearing yesterday within individual organs, as well as earlier today with the nutrition and exercise sessions. So our first speaker for this particular session is going to be Ralph DeBerardinis from UT Southwestern Medical Center.
Thanks very much. And thanks to Jennifer and Rong and Vamsi for inviting me to the meeting. I'm going to talk about the developmental consequences of metabolic defects. And the starting point here is to remind you of the incredible metabolic diversity in humans, much of which is genetically specified. So some aspects of that heterogeneity can cause disease, including developmental anomalies. And if we can trace the root cause of the disease back to a metabolic defect, it presents a great opportunity for discovery. And so that's the theme for the talk. Inborn errors of metabolism are prime examples of this. These are Mendelian diseases caused by enzyme mutations. And here's an example. So two siblings with the same mutation in PAH, the enzyme that converts phenylalanine to tyrosine. And lack of PAH causes the massive accumulation of phenylalanine and related metabolites. So the disease called PKU tells us that PAH is required early in life for brain development because defects result in severe intellectual disability, which you can see in the boy. But the younger sister was treated with a diet low in phenylalanine from birth, and remarkably, she has a normal IQ. So here's an example of an extreme developmental phenotype originating from a mutation in a single enzyme, and the phenotype is mitigated by resetting the metabolic state through diet. So we know of hundreds of inborn errors now, and the question is, why are they still interesting? It's because in most of them, even PKU, we really don't know how the metabolic defect causes the developmental defect. And I think that's an awesome opportunity for developmental biology because the metabolic anomaly is so clearly defined. So how do we study that? Well, first we use metabolic profiling to find outliers in the population and to find the impact of their defects in the metabolic network. And then we try to relate the metabolic anomaly to defects in cells and tissues. The defects are pleiotropic. They involve everything from the death of specific cell types to progressive organ dysfunction to even cancer in some cases. And most of that we don't understand very well. So the state of the field is that we do okay, I think pretty well with the first part, but still pretty poorly on that last crucial part, linking metabolic anomalies to processes in cells that are not intuitively metabolic, but which ultimately determine tissue function. And that's where I think we can make progress. And I'm going to present 4 areas of need. Okay, first, in order to recognize outliers, we have to have a handle, obviously, on the normal ranges for the metabolites that we can detect with broad profiling techniques like metabolomics. And that's an order of magnitude more metabolites than what we detect with conventional testing in the clinic. We've been working on this here in a clinical research program that recruits subjects suspected to have inborn errors. So as an example, we recruited these 2 sisters from Pakistan with a neurological phenotype, collected blood from the girls and their parents for metabolomics, and then compared the levels of each metabolite in the patients, about 400 different metabolites to a cohort of several hundred people to find the outliers. And in this case, the only elevated metabolite was L2-hydroxyglutarate, which is shown there on the right. You can tell that those 2 siblings are obvious outliers for the levels of that metabolite. And exome sequencing then revealed that the girls were homozygous for a nonsense mutation in the L2HGDH gene, and the parents are carriers, as would be expected for an autosomal recessive condition. So those variants impair the function of L2HG dehydrogenase. That's the enzyme that converts L2HG into alpha-ketoglutarate, a Krebs cycle intermediate. And this is a known disease. It's called L2-hydroxyglutaric aciduria. It's autosomal recessive. It causes progressive neurological dysfunction. So we know the cause, but again, we don't know the specific mechanisms by which L2HG interferes with brain development, so we don't have a good way to treat the patients. And we suspect that there's a defect in differentiation because of the parallels with D2HG in cancer, but that hasn't been explored in detail yet in patients. And that's a common theme for these diseases. We know the defect, we know the metabolic consequences, but we don't know the mechanisms of tissue dysfunction. So just a quick point about cellular models. Primary cells have limitations, but sometimes they can be helpful. So here, Wen Gu in the lab reprogrammed fibroblasts from the patients into iPS cells and then edited the mutations to wild type using CRISPR. And that gave him a nice isogenic system to study neuronal differentiation in culture. So the mutations don't cause any obvious problems in the fibroblasts, the iPS cells, or the neuroprogenitor cells, but the mutants have a paucity of these spindly yellow neurites, which are characteristic of differentiated neurons. And the neurite abundance increases when either one or both alleles are corrected wild type. You can see the quantitation there on the right. So that indicates that the mutation creates a subtle autonomous defect in neuronal differentiation, at least in this model. And that may provide a system to screen for manipulations that improve differentiation. Okay, we also really need more effort and high resolution methods to characterize the effects of metabolic dysfunction on tissue architecture and cell-cell interactions, both in mouse models and even in human specimens. This is a surprisingly underexplored area in these diseases where almost everything in the literature is simple pathology from clinical labs. So a case in point, this is an MRI from a baby that has methylmalonic acidemia, a defect in branched chain amino acid degradation. And the disease causes acidosis, which we understand by looking at the metabolic pathway, and chronic defects in the nervous system and elsewhere, which we really don't. Those arrows on the MRI highlight a number of problems with the internal capsule and the corticospinal tract, but there's very little, if any, high resolution data, even in mouse models, to know what's happening at the cellular level in this disease and many others like it. So it would be fascinating to use neural tracing techniques and even single cell sequencing to explore how these how these tissues are altered by the metabolic defect. And that's not unique to the brain or to this disease, but I think the brain is relevant here because of all the tools and expertise in neuroscience at Janelia. So again, the message I want to convey is that we know these metabolic anomalies interfere with tissue function, but we usually don't know how. So we also need better ways to manipulate metabolism in functionally distinct cellular subsets in mice so that we can test regional cell type sufficiency for the phenotype. And I think the liver is a good example of this. Of course, the vast majority of the cells in the liver are hepatocytes, but the hepatocytes are metabolically diverse, as Daniel discussed yesterday. They express different enzymes so that metabolic activities are compartmentalized across the lobule. So to understand metabolic anomalies that manifest in the liver, it would be great to be able to either knock out or knock in mutations in relevant hepatocytes rather than all of them at once. And I think we're getting closer to being able to do that. This is a paper from Hao Zhu's lab that generated 14 different Cree drivers based on zones of expression within the lobule. And what you can see at the bottom are examples of TD tomato expression driven by two distinct promoters in non-overlapping sets of hepatocytes. So we need more models like that, not just for hepatocytes, but for functionally distinct subsets in other tissues as well, the brain, the bone marrow, maybe even different fat depots, et cetera, all of which can be affected by different inborn errors. And then finally, we need ways to understand the consequences of metabolic perturbation during development in utero. And if you were on yesterday, you heard Ashley Salmonson, a postdoc in the lab, talk about her methods to track metabolism in time and location during mid-gestation, even detecting organ-specific fuel preferences and assessing the impact of an inborn error of metabolism, in this case affecting lipolation on pathway activity in the embryo. So I think that's exciting, and I hope we've just scratched the surface on what we can do with these kinds of approaches. Here are some challenges that we need to address in developmental metabolism in the coming years. And the ultimate goal, of course, is to be able to assess metabolic features completely in situ using mass spec imaging and biosensors and perhaps other approaches on intact tissues without the need for dissection. And So in summary, I think a logical approach to explore the impact of metabolism on development is to start with developmental anomalies that we observe in inborn errors and then develop model systems to study them. And I think the 4D cellular physiology program at Janelia could add a significant amount of insight into these efforts, not just through the things that I've already discussed, but also through computational analyses to better extract metabolite data and to integrate that data with other high content data sets, and also possibly ways to multiplex the ability to image multiple different metabolic features simultaneously in the same tissue. Okay, I think that's my time, so I'll stop and just thank the people who generated a little bit of the data that I showed, and I look forward to questions.
Great, thanks, Ralph. That was really terrific. And we have a couple of questions that are coming in. And the first question, really interesting question, can we screen for inborn errors in utero? And would changing the mother's diet during pregnancy help as a part of the child's treatment? Really fascinating question.
It's really interesting. We can't, the practical matter is that we don't have good normal ranges for metabolite levels in utero. There's potentially some interesting opportunities around non-invasive imaging in utero with MR spectroscopy that could potentially be applied. I think the fact of the matter now, though, is that molecular analysis is so far advanced that for patients with known conditions, if there was a condition of suspicion, you would detect this through, a sequencing in chorionic villus sampling or an amniocytes. Does affecting the diet of the mother impact the metabolism of the patient? I can't think of a great precedent right now for where that would mitigate the phenotype, but there surely is impact of the maternal diet on metabolism of the fetus, and then also postnatally. So there's a number of diseases that are mild enough that the mother can have the disease and not be diagnosed, and then the child will flag the newborn screen for the abundance of some metabolite that actually crossed over either from the maternal circulation or in the breast milk. Gestational diabetes, of course, is another non-Mendelian example of this, where the maternal metabolism has major impact, even teratogenic impact on the fetus. So that's another example.
That was a great question from Danielle Smith, and I guess folate supplementation would sort of fall in the spirit of that as well.
That's great.
For rare and for common, probably.
Of course, thank you very much. Right, so mitigating neural tube defects, very important.
So another question from Chalson Zhang. Why is the brain mostly affected by inborn errors of metabolism despite the blood-brain barrier that may be more selective for metabolite trafficking than other organs?
Yeah, I think the metabolic needs of the brain, of course, are very high in terms of energy metabolism. I think that's probably why many of the energy creating pathways, if they're defective, manifest in the brain. I wouldn't say the brain is necessarily the most commonly affected. We see lots of examples of inborn errors that will manifest in the liver or the muscle, but not the brain, but relatively brain sparing. And so I think it really, there's a lot to be learned there, actually, by which which defects and which pathways manifest in which tissues.
We have a question from Luke.
Hi, Ralph. Very interesting, the L2HG stuff. You must be thinking about epigenetic change there, right? Because the L2HG could affect demethylases, couldn't it? It'd be great to look at the epigenome, wouldn't it? Yeah,
that's right. So thank you, Luke. All the story about D2HG and cancer, and so that was why we started this in the first place. The problem with L2HGDH deficiencies is we just don't have good cellular models that have a phenotype, right? We've had fibroblasts in these patients for a long time, but they're basically healthy. So identifying a context that can be manipulated in culture that creates a defect, that now we have opportunities for discovery. So we've done RNA sequencing already, and we have a number of genes that are altered in the neurons that are trying to differentiate, and some that are even altered in the pre-existing neural stem cell state.
Just looking for other questions. I think your student has a question, so Ashley.
I do. I wonder if you could comment on some stuff that Jared talked about yesterday. So he talked about cycles between different cell types, like in the brain, lactate shuttles and whatnot. And what are your thoughts about that in relation to tissue specificity when it comes to inborn errors?
I think this is a vital question, and I'm ashamed that I didn't plant you to ask that question, but this is exactly the reason why we want to be able to create metabolic aberrations in distinct cell types, right? Like, I mean, there are cases of inborn errors that do manifest in the brain where, you know, we were all sure that the primary problem was in neurons, and then you make a neuronal knockout and the mouse has no phenotype, you knock it out in the glial cells, and then there's a prominent phenotype. So, you know, this kind of thing is probably playing out all the time in tissues, even when we think the cells should be homogeneous, again, with metabolic zonation among hepatocytes being an example of that. And so to understand these interactions, we have to be able to go in and surgically knock out or knock in mutations in exactly the right cellular subset.
Great. Well, thanks, Ralph. That was a really terrific talk, and we'll continue the conversation in the group discussion. So now we're going to shift a little bit from development to the other extreme aging, and we're going to hear from Toren Finkel from the University of Pittsburgh.
Thanks very much to the organizers for this opportunity. As Vamsi said, I'm going to switch to the other spectrum of biology here and try to talk a little bit about the relationship between aging and metabolism. I think the canonical sort of link between these two phenomena has actually been appreciated for quite a long time. And so actually over 500 years ago, Luigi Cornaro, who was a Venetian nobleman, was about 40 years old and was told by his doctors he didn't have much longer to live, and decided he would change his lifestyle and went to gave up a lot of things that he was indulging into, but one of those was food. And for the next almost 60 years, until his death at age 98, he ate a very sparing diet of about 350 grams of basically soup, bread, and egg yolk. Since he was Italian, he also included 2 glasses of wine every day as well. And when he was about 80 or 85, people were very impressed by his longevity, and he decided to write down his secrets. And he wrote probably the first sort of medieval bestseller on diet, which he called Discourses on the Sober Life, which was a big hit in the Middle Ages, detailing, you know, the benefits of reducing food intake in terms of longevity and vitality. And that was actually ignored in the scientific community for a while, until Clive McKay's group at Cornell, roughly about 100 years ago, did the first sort of controlled experiment in rats, where they restricted food intake, but not to the point of malnutrition, but restricted food intake to rats, and found that those rats grew up to be smaller but long-lived. And that really has formed the basis for a lot of observations in both rodents and other organisms, which has been reproduced in many, many labs and in many, many conditions, demonstrating this very interesting link between how much we eat and how long we live. And although this has been, in one way, described for the last 500 or so years, remarkably, there's still a lot of mystery as to about how this happens. Not that there hasn't been progress, and one of the sort of certainly links between food intake, food availability, and lifespan comes through a pathway that we've already heard quite a bit about, which is the mTOR pathway. And there's a significant amount of evidence that suggests that reducing mTOR activity, which mimics reduction in food availability, extends the lifespan of organisms. And shown on the right is work from our lab a few years ago, which reduced mTOR levels to about 25% of wild type, and that resulted, in the case of mice, in an extension of lifespan of about 20%. And similar increases in lifespan has been shown with pharmacological inhibitors of mTOR, as well as other genetic strategies to reduce mTOR activity, again, in a wide range of organisms. But It's certainly not the entire answer, because if you look in lower organisms, for instance, at strategies that reduce mTOR activity and prolong life, you can still get additional benefits from reducing caloric intake in those model systems, suggesting that not all the effects of mTOR, of caloric restriction are mediated through a reduction in mTOR activity. And in fact, changes in food availability, caloric restriction produces a wide array really of metabolic and physiological effects. Some of those are shown here. One thing it doesn't do is to change metabolic rate. And we'll come back at the very end at this sort of interesting relationship between metabolic rate of an organism and its lifespan. But caloric restriction by itself does not really affect metabolic rate, but it does affect a variety of other metabolic parameters, as well as affecting important physiological parameters such as metabolic flexibility, the ability to switch between carbohydrates and fat as a source of energy, and a variety of processes. And together, these integrate in some fashion to an increase in overall lifespan. But again, the exact metabolic process, the exact metabolite, and the tissue in which this is occurring is really not very well understood, and certainly not in humans. And the question is, why, for a phenomenon that's been known in some regards for 500 years, have we not made a significant progress in understanding the mechanisms involved? And I think I wanted to return to a theme that was brought up yesterday, which was, the difference between metabolic research and metabolism research and molecular biological research, and the inability to use gain and loss of function tools currently in metabolism to the same degree of effects that one can do in genetic knockdowns or overexpression studies. And I wanted to give 3 quick examples of work that we've done, or some of my colleagues at our institute have done, that sort of try to address this idea of tuning metabolites as a way of understanding their function. So individual changes in metabolites within cells or tissues as a way of trying to deconvolute how something like caloric restriction might extend lifespan. And the first example is done by a colleague of mine, Yosuke Sukain, in our institute, who was interested in manipulating specifically acetyl-CoA levels in cells. And this is just an example of how the approach that he took. And so, as some of you know, acetyl-CoA levels in the mitochondria and acetyl-CoA levels in the cytosol and nucleus really represent 2 pools of the metabolite that are not in equilibrium. The majority of cytosolic acetyl-CoA is generated by the action of the conversion of citrate by the ATP citrate lyase enzyme in the cytosol to form acetyl-CoA. And that again produces the bulk of acetyl-CoA that's then used for a variety of modifications of proteins and for the synthesis of lipids. And in fact, it's actually quite hard in many cells to knock out ACLY by CRISPR, just because of the essential role of that enzyme in generating acetyl-CoA in cells. And so what Yosuke found was that he could actually, in cells that you were unable to knock out ACLY out, you could do that if you supplemented the media with high levels of acetate. And that's because there's another pathway for acetyl-CoA generation where it uses exogenous or endogenous acetate through the actions of enzymes such as ACSS2 that converts acetate into acetyl-CoA. And so what he was able to do is, in the presence of acetate, exogenous acetate in the extracellular fluid, he was able to generate cells lacking ACLY. And those cells then became sort of auxotrophic for exogenous acetate. And so if you removed acetate from these cells, they had a rapid fall in intracellular cytosolic acetyl-CoA levels in those cells. And so it became a way, just by growing cells in the presence of acetate and then removing it, one could rapidly deplete in a very specific way intracellular acetyl-CoA. And what he observed is that there was in fact a rapid mobilization of rapid deacetylation of both histone and non-histone proteins under those conditions within sort of minutes to hours of removal of acetate. And that's sort of an interesting, and so that became sort of a biochemical way, in this case, to specifically regulate one specific metabolite in a cell. And it sort of suggests that, you know, an interesting concept perhaps that there's sort of evidence that many acetylation marks and other epigenetic marks don't appear to regulate transcription of neighboring genes. And this rapid mobilization of acetyl groups in the setting of nutrient stress suggests that maybe another role for modifications of proteins, and they may in one way serve as depots for for metabolic intermediates, and may be utilized and actually liberated in times of metabolic stress in cells. A second thing that was brought up yesterday is the use of pharmacological agents to tune and regulate metabolites. And so we've been interested in NAD metabolism in cells. And as some of you know, that the NAD formation in cells is complicated, but the predominant enzyme, the rate-limiting enzyme in the NAD salvage pathway is an enzyme NAMPT. And so with my colleague Bill Chen, we've been interested in generating pharmacological tools that both inhibit NAMPT and thereby lower NAD levels, as well as pharmacological tools that actually bind to and activate NAMPT and thereby increase NAD levels. And as you can see here are a series of compounds that we've generated that have different efficacies, but allow us, using a pharmacological agent, to manipulate NAD levels by, again, using a small molecule to regulate the activity of rate-limiting enzymes in a certain biosynthetic pathway. And so these might be useful to dissect out how NAD might be useful in modulating some of the effects of caloric restriction. And finally, I wanted to just give an example from another colleague of ours that did work that he did prior to joining us in Norbert Perman's lab, Andrei Parhitko, who was interested in this idea of methionine restriction as a means of extending lifespan. And so again, just like caloric restriction, people have played around with diets, and diets poor or absent in methionine, as shown here on the left, are able to extend the lifespans, again, of rodent species. And again, how this is working or where this is working has not been that clear. And so what Andre did while working in the Perman lab was to employ a bacterial enzyme methionase, which is able to degrade methionine into ammonia and other components. And in data that I won't show, he was able to show that he could express this in animals in a tissue-specific fashion and lower the level of methionine in that tissue to levels that were similar to what one sees in a methionine-free diet in flies. And then shown on the bottom here is the expression of that construct of methionase in the brain of flies, which recapitulated the life-extending effects of methionine-free diet, suggesting that a reduction of methionine in the brain was sufficient to extend life of these animals. But again, this provides, in this case, the use of a bacterial enzyme, which is similar to what Vamsi's also done in other cases in enzymes that regulate NAD metabolism, but using bacterial enzymes to manipulate in a specific, in this case, tissue-specific fashion, individual metabolites. And again, it suggests, based on Andre's work and Vamsi's work, that we can maybe leverage some of the enzymes in bacteria to understand mammalian physiology or other animals' physiology, and sort of try to dissect, which are the important metabolites and where they are acting. With that said, I just wanted to sort of shift and say that as important as these model systems are, and as interesting as they are to manipulate, I think there are some caveats in terms of our own physiology. And I just wanted to highlight again another one of my colleagues' work that was done before he joined us in Boston. And this is Matt Steinhauser and his wife, who were both up at the Brigham and the Mass General. And Matt actually took people at the Brigham, volunteers, and starved them over a 14-day period on a 0-calorie diet. I think what's interesting, and in this case, he looked at the response of those individuals in many ways, but I just wanted to highlight one sort of interesting difference between mice and humans. On the left, you see the induction of one of the sort of mitochondrial stress hormones, FGF21, which in mice rapidly rises after fasting. So by 6 hours, there's already a 4 or 5-fold induction in circulating FGF21 that goes to about 8-fold after a 24-hour period. And in the middle, you see the same same level of FGF21 in humans, again, subject to this, in this case, 10-day fasting paradigm. So again, 0 calories over 10 days. And what you can see is that really until about day 7 or 8, there's really no induction in FGF21 levels in humans, whereas opposed to mice, this happened in a relatively rapid fashion. And again, there's many reasons that this could happen, but I just, the difference that we've talked a little bit about this between small animals and large animals is that, mice in an overnight fast in the paradigm that gave rise to this induction of FGF21 in Matt's lab, they lost about 17% of their body weight overnight. And humans, over this 10 days, lost, at most under 10%. So there's a huge difference. Obviously, if we could lose 17% of our body weight after an overnight fast, the obesity epidemic in the U.S. would be significantly different than it was. And so, this really comes down to, are all these data that we've generated in animal models relevant to humans? And does caloric restriction, will it have the same benefit in us as it does in mice? And so in mice and rats, that benefit is quite large. So you can get a 40% or 50% increase in overall lifespan in the calorically restricted animals compared to the ad libitum animals. In contrast, here's some data from 2 groups that have been doing a 30-year-long experiment in macaques and non-human primates, looking at caloric restriction. One done at the University of Wisconsin, the other one done at the NIA in Baltimore. And they've come to somewhat different conclusions. The Wisconsin group said that there is a benefit in their hands for caloric restriction, while the group at NIA has not really found such a benefit. And there's differences in the paradigms that were used and when caloric restriction was initiated in these 2 different experimental venues. But I think it's also fair to say that the magnitude of effect, even in the Wisconsin group, is significantly less than what we've seen in mice. Again, suggesting that the benefits of caloric restriction may be very different in small animals that have a very short reproductive period compared to animals such as humans, in which there's a much different physiology, and some of that I've tried to highlight. So I just want to end a little bit coming back to this idea of metabolic rate and lifespan. And again, this is an interesting correlation that's been made that's been known again for about 30 years, since Raymond Pearl sort of described what he called the rate of living hypothesis, which suggests that there is this inverse correlation between metabolic rate and overall lifespan. And not every organism, including humans, don't fall on this curve directly, suggesting that it's not a perfect explanation. And as I mentioned before, interventions like caloric restriction, which extend lifespan, don't appear to affect metabolic rate. So it really doesn't suggest that metabolic rate per se is what's driving lifespan, but something linked to metabolic rate appears to be important for how long we live. And so one idea that I proposed a few years ago, which I think is sort of interesting, and it's sort of interesting maybe to think about for Genelia, is not necessarily metabolic rate, but sort of energy partition, which, again, a lot of metabolic research is trying to think about how we generate ATP, what pathways we use, but very little is given into the thought about, once that ATP is generated, how the cell or tissue or the whole organism decides to use that ATP and what it directs that ATP for. And so shown here is an example of, how, on the left, a pie chart of how an organism might, partition its energy.
Hey, Toren, I'm really so sorry to interrupt, but we are so well over time at this point.
Okay, so
we have to ask that you conclude, and then hopefully we can circle back to some of your points in the discussion. Thank
That's
you.
it. I'll stop. Sorry. Thanks very much.
Thanks, Toren. And there are some questions for sure for your very provocative talk, but we'll get back to those in the Q&A. And so our next speaker in this session is Dr. Marc Kirschner from Harvard Medical School Department of Systems Biology. Marc. Marc, you're on mute right now.
All right, I'm unmuted, but I'm still having a problem sharing my screen.
I think if you just go into presentation, then we should be okay. We can see.
Stop share, it says. You're sharing your screen, it says. Is it sharing or not?
Yeah, we can see it, Marc. I think if you just
I'll go to presentation. Okay, I can't see. Fine, great. Okay, thanks. So, sorry about that. First of all, I want to thank everybody for inviting me. Everybody knows that metabolism is important. Not everybody knows it's so interesting. So I think we have something we need to do here in terms of publicity. So first of all, nutrition affects every level of biology, and I think we saw very good evidence of that through this talk. But the place where most of biology has been worked out, really, at least in the last 150 years, has been at the level of cells. And I think there's much more to be done here. That's what I really want to talk about that. Cells respond to nutrition. Actually, they're pretty resistant to responding to nutrition. If you try to inhibit their growth, they tend to remain more or less the same size. They may slow down a little bit, but cell size, as an example, as a function, is well maintained under very nutritional conditions. There's an impressive constancy of mean cell size in proliferating populations. And yet we know that under certain conditions, there's hypertrophy, such as cell differentiation, senescence, and disease, and we don't know how hypertrophy comes about. I would argue that one of the problems we have with cells, which is probably not appreciated, since we have electron microscopy and immunofluorescence, is the ability to accurately measure cell mass. And it would be nice to separate that into proteins and lipids and even further. And so what I really am going to talk about is what that opens up. First of all, let's talk about proliferating cells, which is really a problem, which is quite interesting. There must be some sort of feedback on size and growth, because we know that cells actually grow to some degree in proportional to size. They're not growing independent of size. And if that were the true, then any slightly larger cell would grow faster. And there would have to be some sort of feedback. And just to show you an example of what this looks like, here's just a model of situation with linear growth where there's no dependence on size. And the population maintains its distribution very well. If you have a dependence on size, then it starts out looking pretty much the same, but very quickly, the variance in cell size increases indefinitely. And so this is a problem that most of you haven't thought much about, but it's something the cells have figured out how to resolve. That is, how to know how large they are and how to retain their growth if they're too large, or how to increase their growth rate if they're too small. so a lot of what I'm going to talk about depends on new methodology. This is a new method based on a method actually that was developed by several other people, including somebody who's now in my lab, but called quantitative phase microscopy, and she developed computer enhancement to this. And here's a dry mass of a cell or a group of population of cells as a function of time. Nothing very extraordinary here. Here it is sort of, since we're looking at it by microscopy, we can superimpose it, these cells, so that we look at their time since division. We don't see much there. But with the increased precision of these methods, we begin to see, if we look at growth rate, some hints of oscillation as a function of time. And then if we look at really the derivative of that, we see a very distinct 5-hour oscillation in growth rate, which is initiated by cell division, but which doesn't correspond to any other transition that we know in the cell. And it might very well be that this is some sort of metabolic oscillation. We've seen this now in multiple cultured cells, both attached and unattached cells. And this shows you, when you begin to look closely at things with improved methodology, we begin to see phenomena which we now want to understand, because it may tell us something about how cell cycle is controlled, how cell growth is controlled and how it's affected by size. Well, what we all would like is a method for looking at protein, lipid, RNA mass accurately, deep in fixed or even in living tissues, quantitatively, at great depth, and without suffering minimal scattering. And the approach that we worked out started with some help from Sunny G's lab, but it's really the work of a very talented PhD student from MIT in optical physics, and who's been in my lab and has worked on this for quite several years now, is to use Raman microscopy. And for those of you who have some memory of chemistry, which is probably as weak as my memory of metabolism, the Raman microscopy is a method that looks at scattering. It's quantized scattering. I won't go into detail. And it obviously is sensitive to the chemistry of the cell, and it's particularly chemistry to easily distinguish between protein and lipid, looking at the methylene hydrogen groups on the fatty acids, or looking at the methyl groups on things like isoleucine and valine. And it's a very imprecise method, because a lot of things that will give you change the value. But she figured out that including water in the specimen, that is measuring both at the protein wavelength and at the lipid wavelength, but actually including water, that you can use spectral decomposition by using standards first and then go back into cells. And this has a dramatic effect of allowing very deep measurements into cells. That is, living cells, we can get down to about 150 microns into cells, and I'll show you beautiful images. And if you do some special treatments in fixed cells, you can get down as deep as a millimeter, many, many, many cell layers, and look quantitatively at the mass distribution in different parts of the cell and the lipid distribution in different parts of the cell. So let me just give you a couple of examples of this, of different cells, and showing how different lipid density is. And in this case, looking at just a single section, but we can reconstruct them in 3 dimensions. And looking at protein density, and I won't go through all of these, but you can see quite clearly that in the case of the kidney glomerulus, the tubules and the glomerulus rather are in the kidney are quite different in their lipid density for sure, and even in their somewhat in their protein density. And we look at it in other tissues like liver or skeletal muscle. And these are really typical type images that you would get from this. And as I say, you can do 3-dimensional reconstruction and calculate the total mass as well. and the total of proteins and lipids, and distinguish things like the composition of saturated, unsaturated lipids. Also, we've done nucleic acids as well. Now, typically, I mean, very commonly, I should say, hypertrophy occurs with constant protein density. So protein density seems to be something that's actively maintained. Here we see cultured megakaryocytes that have increased dramatically in size without increasing their protein density. We measure the dry mass and volume are linearly related. But of course, in other types of tissues, protein density is part of the differentiation process, typically in bone, where we can see the change in dry mass density. This is now looking at protein plus lipid, and it's different for different types of bone as they grow. The difference between the radius and the tibia have different rates and times of hypertrophy here, which are really mostly swelling and some growth in mass. So this feature of mass and volume is fairly important. A lot has been said about cell dilution and senescence. A lot of it, I think, is wrong and is due to poor measurement, and I don't want to, but you can see when you look at lipid as well as protein, you see quite different things. For example, in the MDCK cells, which are induced to senesce, you see a drop in protein concentration, as shown on the left here. But you also see a very substantial increase in lipid. Whereas in melanoma cells in culture, there's a smaller drop in protein concentration and a bigger increase in lipid. And we want to look at these things in obviously aging animals and aging humans, and not just in model systems. And we've begun to do that. So this is my conclusion here, is that like virtually everything in biology, understanding comes at the level of the cell, or a lot of understanding, not complete understanding. Knowledge of chemical composition. So the concept of cell size is a very vague one. It's also poorly measured. And we need to think about protein mass and mass density and lipid mass and mass density and ionic composition. And also we need to have methods to look at the spatial distribution at the highest possible resolution. And we need to understand something about the changes that occur both in the cell cycle, as I showed you, where we get these very dramatic oscillations of low amplitude, but very specific repetitive frequency. And that leads to models that we can make and that we can show to be invalid. And then, of course, when we have these things, we can perturb these things. We can perturb them genetically. We can perturb them by perturbing metabolism. We can perturb other specific enzymatic activities like protein kinases. And the other aspect, which I don't have time to go into, but clearly the set points for protein density, protein mass, that also has changed in evolution. And I think as we look at the differences between different animals, even different mammals, even say mice and humans, they may give us certain insights into how cells maintain their size, but also how cells change during evolution and differentiation, and also how to interpret some of the perturbations that we add to that. So thank you.
Great. Thanks, Marc. See, I'm going to begin with one question. You showed with these really careful measurements these oscillations and are speculating that they may be metabolism related. And I'm wondering, what are your strategies for trying to figure out what the metabolic link is? And number two, might this be related to some of the redox metabolic cycles that have been described by Ben Tu and Steve McKnight?
They might be, but those were generated in a almost a forced condition that they created. And if so, then that would be very interesting, because I don't think anyone's seen anything like this here. I mean, our first goal is we can look at mass and lipids separately. Of course, when we're looking at protein density or protein mass, we want to know, is there an oscillation in protein synthesis or in protein degradation? And I think in general, that's a big starting point for us to be looking at it, because we can then look further at where that arises. Then, I mean, we would love to follow that up with looking at metabolic things that might be regulating those things. But we're going to take it one at a time we don't have a favorite model that we're going to go after to start with.
Great. Meng?
Marc, it's great to hear another person using ramen technique.
Yes.
Yeah, so I think quick question. So you are using the CH region or using the fingerprint region for those images?
Are we using what?
The CH region for protein lipid, or you are using the fingerprint region?
We're using the CH3 region.
I see.
For protein. And I should say one other little thing. As you probably know, we can look at protein synthesis and degradation in living cells, because by feeding them deuterated amino acids, we can actually measure the incorporation into protein or the loss in real time. And I think that has other applications.
Yeah, it's great. Yeah, because I think, they're technique ready for the fingerprint region, and definitely we can recover more chemical features there. I think be even more exciting. Yeah, for
very
future.
good.
Yeah, thank you.
A question from Bruce.
Hey, Marc. So one of the applications that could be really exciting is there's a huge paradox in hypertrophy in cardiomyocytes.
Yeah.
It looks very superficially similar, malignant hypertrophy and beneficial so-called physiological hypertrophy. Athletes, not just do their hearts get bigger, the cells get bigger. And the same thing is true, unfortunately, in pathological hypertrophy. So superficially, it looks like in both cases, the cells get bigger. But what is actually going on at the level of analysis that you are trying to push? They get bigger. That's all people really know. But one is leading to heart failure and one is leading to athletic performance. Could you get in there and actually look at the cardiomyocytes under these two very different, they look the same, but they give very different consequences?
It would be very easy to do that. And I should say, when people say the cells are bigger, they have to be very much bigger for them to really say they're bigger.
Yeah.
And here we can look at changes in cell mass in tissues at the sort of 1% level. And so that, I think, will allow us to look. Those may be the dramatic examples, but there may be other diseases, other conditions where the effects are much smaller and may not have even been.
But this can be modeled in mice by forcing mice to run or swim on the one hand versus using transaortic constriction. And you end up with big cardiomyocytes in both cases.
That sounds quite cruel, Bruce,
The running or the transaortic constriction?
The running, because I don't have a lot of trouble. But anyways, no, I think it would be terrific. I wish we should really plan to do so. Somebody should plan to do that. There's only so much we can handle at once, but we're definitely interested in the question of cell size and hypertrophy generally, not just in aging, but in other types of disease conditions.
Great. I know that there's more questions in the chat box as well as from Ralph and from Jennifer, but we're gonna have to save those for the group discussion. So thank you very much, Marc, and we'll have those questions for you in a few minutes. So our final speaker for this particular session is Karin Ortmayr, who's going to be talking about metabolism and quiescence. So Karin.
Yes, thank you very much. And thanks to the organizers. Let me just check the screen. Yes. For selecting my abstract and giving me the chance to speak here at this very exciting meeting. It has been very stimulating already so far. So I want to use the opportunity to tell you a bit about my postdoctoral work in the lab of Zampieri at ETH Zurich in Switzerland. And my work has been evolving around understanding the role of metabolism in cellular quiescence. And so quiescence, just a bit of a description in a nutshell, if you want. Quiescence is a quite specialized cell state for actually many different types of human cells. And it is characterized by these cells undergoing a growth arrest, but it's not a terminal growth arrest. This is reversible. And it's the key characteristic of quiescence. These cells can survive for very long times in this non-dividing state and yet are able to return to proliferation quite spontaneously given the right environment, in short. And this makes quiescence very important aspect of many aspects of human health, including immune system. But also in recent years, we've increasingly understood that quiescence also plays a role in some diseases, including cancer. And in particular, it is probably involved in cancer recurrence and metastasis. And so more work has been done on identifying, and there have been reports that small subpopulations of cancer cells can exist in this quiescent state in vivo in the patient. They can survive the cancer treatment and remain almost undetected for long periods. And after those times can still give rise to a renewed tumor growth, which is then called cancer recurrence. And this is actually quite big and it's sort of long-term issue in clinics and is still unsolved. And I think in part this comes a bit from the fact that for long years, the primary perspective on cancer cells was the one of rapidly dividing cells or proliferating cells that are really needed to sustain this very fast tumor growth. And this has been extremely successful research perspective, if you want, because it has uncovered many key aspects that were eventually implemented in treatments, including the fact that metabolism is very characteristically changed in cancer cells and actually is crucial for these cells to sustain this rapid growth. In contrast, for other cancer cell subpopulations, such as non-dividing cancer cells, relatively little is known in general, and in particular about the role of metabolism in allowing these cells to undergo cell cycle arrest, maintain homeostasis, and yet be able to return to proliferation at a later point. And so these are a bit the questions that we're after here. What is the role of metabolism in helping these non-dividing cancer cells, more from a fundamental perspective, but also can we find essential metabolic processes that might be actually useful in treating these very difficult to target cancer cell subpopulations. And so here we focus on environmentally induced quiescence. So there's environmental stimuli that are also found by these cells in vivo that we can apply in vitro to stimulate quiescence. These include the absence of growth factors, spatial constraints, and also nutrient restriction in general. We can use these environments on here 6 different cell types because we were really aiming at finding metabolic characteristics of these cell types that are not specific to a particular cell type, but are more conserved. And so what we found here, applying these environments, these 3 different ones to the 6 different cell types, we found that in all conditions, we could actually enrich the population for quiescent cells. This G0 phase as assayed by flow cytometry-based assay. And this was an important first finding in that the enrichment went up to 30 to 60% of the population in 4 days. This means actually that this model sort of recapitulates that also in vivo, these quiescent cells, they don't really occur as a pure population, but they coexist with cells in other cell cycle phases and states. And so because we here were particularly interested in understanding the specific metabolic profile of G0 cells, we came up with a solution that avoided having to physically separate out the G0 cells from the population. And instead we could leave them there and instead use a combined experimental computational framework that allowed us separating the metabolic signatures of these 2 subpopulations without cell sorting, which can induce severe metabolic artifacts. For sake of time, I won't go into much detail, but I'm happy to take any questions on that later on. The important message here is this methodology allowed us to actually systematically study the metabolic differences between quiescent cells in G0 and cells in other cell cycle phases in all of our 3 environments and 6 different cell types. And with this data in hand, we were able to extract a characteristic signature of metabolic changes that were conserved across all of these 18 different models. And these are depicted here in this volcano plot. So you can see we found a set of metabolites on the left-hand side that were consistently at a lower abundance in G0, while on the right-hand side, a very interesting set of metabolites was consistently more abundant in cells in G0. And we actually ended up focusing a lot on these accumulated metabolites because they consistently indicated a signature of a characteristic change in lipid metabolism in G0 cells. More precisely, they indicated or led us to the hypothesis that fatty acid beta oxidation, the perhaps major pathway for the degradation of fatty acids in cells, seems to be characteristically altered. And in following, we pursued the idea that this could actually be a key metabolic hub in quiescent cells and could be linked in many ways to survival and homeostasis. And so we started investigating the role of fatty acid beta oxidation in quiescence using a pharmacological inhibitor, that's trimeticidine, an approved drug that inhibits the last enzymatic step of beta oxidation, where the acyl chain is actually shortened. And so we applied this drug in quiescence-inducing or promoting environments, and then followed the growth or cell confluence, as it's shown here, during quiescence induction. But we also wanted to see what the effect is on the ability of these cells to return to proliferation. And we tested a wide range of different doses of trimeticidine, and we consistently saw, very interestingly, that both the induction of quiescence or the transition from proliferation to quiescence was inhibited with trimeticidine, as well was the regrowth, sort of the time at which the cells were returning to proliferation to show the normal growth rate, if you want, was greatly delayed thanks to trimeticidine. So this was very intriguing finding that we may have found a metabolic mode that is really essential in this quiescence-inducing environment. And this became even more evident when we looked at the effects on viability, not only on the growth inhibition. And what we saw is that across the entire concentration range that we tested, we saw a dose-dependent reduction in viability, and that trimeticidine really induced cell death in these cell populations. But this was really limited or selected in the quiescence-induced environment. In full growth medium, where the cells rapidly proliferate, there was absolutely no effect of this drug on viability, and minor, if at all, any effect on the growth rate. So taking this together, it really supports the fact that fatty acid degradation and its enhancement, if you want, is characteristic for cells in G0. And so we wanted to understand in more detail what is the toxicity mechanism underlying this inhibition of beta oxidation. And we looked at the metabolic consequences of this fatty acid beta oxidation inhibition by trimeticidine to understand more. And to keep it really short, what we observed is that a number of lipid-related metabolites accumulated, and most notably several ceramides were among those. And I'm highlighting those because ceramides are actually known bioactive lipid species that are perhaps most appreciated for their role in inducing apoptosis. To support such a link that perhaps trimeticidine induces ceramide accumulation and thus induces apoptosis, we found that the ceramide accumulation was strongly correlated with the cell death in the population. We were eventually also able to confirm that indeed these cells undergo apoptosis treated with trimeticidine using a fluorescence imaging assay that tracks markers of early apoptosis to the exposure of phosphatidylserine residues on the cell surface. We used a dynamic imaging assay here, and we were able to track over time that early on during the treatment and the transition, we could see the emergence and steep increase in the number of early apoptotic cells, followed by a rapid loss of membrane integrity and massive cell death by the time of 96 hours as seen before. And this same pattern we saw in different cell lines at different doses of trimeticidine, really supporting the mechanism within that trimeticidine, by inhibiting beta oxidation, probably perturbs the lipid balance in the cells and eventually leads to the formation of ceramides that then induce apoptosis. And again, according to our data, this is really selective in quiescence-promoting environments, as the induction of apoptosis was completely absent in normal growth media. So that finally brings me to the conclusion. I've showed you how we used a new approach for the sorting-free metabolome profiling in cell subpopulations, which here was really instrumental to cleanly identify a characteristic and conserved metabolic signature in multiple quiescent cell models, and the metabolic adaptation to quiescence, this long-term non-dividing cell state. And I've also shown you that we can find parts of this metabolic adaptation that are essential, such that this transition into quiescence actually uncovered new and selective metabolic dependencies that we could use pharmacologically to target some cancer cell subpopulations that are currently difficult to treat. And I think what our results call for is perhaps a change in perspective, that a drug like trimethoprim we would not have found in a classical anti-cancer drug screening, because we typically use proliferating cancer cell models for that. So if we consider systematically cell subpopulations and environmental stimuli and their effect on the metabolic adaptation and profile, then we have a better shot at finding really new approaches and combination treatments that could enhance conventional therapeutic strategies and provide a better outcome of the treatments. However, at the moment, this is a bit limited, and I completely agree with what Ralph said. We need better models and a better mapping of subsets of cells and better tracking methods to really manipulate these cells also in vivo models, to really take this to the clinics if you want. Lastly, let me briefly thank the team in the Zampieri lab and our companion labs at ETH, led by Uwe Sauer and Nicola Zamboy. And I'm happy to take any questions.
Great, thank you, Karin, for that great talk. I think we have time for a few questions before we go to the group discussion. I'll start with one. You looked at a couple of different cell types, if I'm not mistaken, some of them were primary as well as cancer. And I'm wondering, if you look at the metabolic signature of the quiescence for the cancer versus normal, are there any notable differences?
Yeah, so yes, we had 2 cell lines in there that were fibroblast cell lines and non-transformed. I don't think we have the statistical power, if you want, in this subset to really say, make a very strong statement about the different metabolic signatures. Here we specifically looked for conserved metabolic responses across all of them. So at the moment, I think one would have to expand this subset of non-transformed non-transformed cells to really make a stronger point about these clear differences.
We have a question coming in the chat box from Hosung Bai. Great talk, Karin. Would you be able to apply your sorting-free metabolome profiling to tissue samples?
Yes, so the key aspect is that we need to know how many cells of which cell population are in there. That's the most deciding. So you need to have a clear handle on how to assign cells to a subpopulation. That's actually the key ingredient and the key limitation, yes.
Ralph?
Karin, that was really interesting. Do you know, does the mechanism of activated beta oxidation involve an increase in CPT1 expression? Because if so, you could potentially then go looking for a change in CPT1 expression in some of the mouse models of dormant metastasis. Whereas I think right now it'd be hard to do metabolic profiling in those models.
Absolutely. So we have actually tested, but it's interesting what the role of CPT1 is actually. So we have also used an inhibitor of CPT1 to understand whether it's mitochondrial metabolism that's involved. Is a bit of a side story if you want. But what we found is that actually CPT1 inhibition can rescue partially the viability of these cells. So it seems to be that that CPT1 is indeed a key factor. And we have seen a surprisingly low sensitivity to CPT1 on its own, where I think the cells can compensate by perhaps using peroxisomal beta oxidation. So it's an interesting question whether perhaps the CPT1 level is so enhanced that we would have to go to extremely high concentrations of the inhibitor. That's how much I can say about the higher levels of CPT1A or not. It could be. We haven't specifically tested it.
Thank you.
Luke?
Karin, that was really interesting. Well done. Fascinating stuff.
Thank you.
I'm wondering about I saw taconite, my favorite metabolite at the moment. Taconite and citrullinite were up, weren't they? So I'm wondering what's happening to oxphos, because taconite can block STH, for example, you know? It can also do many things. That's one thing. So what's oxphos like in the quiescent cells? I guess it's been measured for decades, mind you. Secondly, if you added taconite, would you drive quiescence, I wonder?
So we haven't specifically tested anything around itaconate. It could be, so we see a strong accumulation of aconitate, which is one of the metabolites that was strongly accumulated. It's downstream of citrate, which we would think is the major product of beta oxidation in these cells. And then itaconate, as far as I understand, is a downstream product of aconitate. So it could be that it's more of a passive effect. Whether it also has a regulatory role, we haven't specifically tested, but it's an interesting possibility, yes.
Yes, citraconiate is very interesting as well. That's an unusual isomer of taconite, which is rarely being reported, actually. That's a very electrophilic compound and can react with multiple targets, I guess. It's just a fascination. You've seen those two go up. I think it's well worth pursuing those further.
Thanks.
We'll take one final quick question from Jennifer and hopefully a quick answer from Karin.
Yes, Karin, great talk. You had 3 different conditions that you used for looking at conversion to quiescence: serum starvation, contact inhibition, glutamine inhibition limitation. And I'm curious as to whether you tested all 3 in terms of the effect of the trimetazine, and whether there were differences in terms of the extent of apoptosis that you found.
So the apoptosis we've only tested in serum starvation because it's one of the most robust ways of inducing quiescence. We've also tested a lot in glutamine limitation, not specifically apoptosis, but in general, and we see that the cell phenotype is very different. So we see very large droplets that we don't yet have a clear idea what they are, emerging very quickly within hours in these glutamine limited cells. So that could be some more obvious crosstalk, if you want, with mitochondrial metabolism.
Do they undergo apoptosis? Do you see
we didn't check that specifically. They for sure die. This much I can say.
Okay, but you didn't look at the extent. Okay, cool. And contact inhibition you haven't tried?
And no, because many of our approaches rely on time-lapse microscopy, and their basically contact inhibition is difficult to treat. That's why we didn't include it so far.
Great. Thanks.
Good. So thank you very much, Karin. That was terrific. So now I'm going to try to open it up now for a discussion for the entire group. So this is a great session because we went from development to aging. We talked about growth, and we went from quiescence to senescence, but not in that order. So I'll sort of open it up now for broader discussion. And Meng?
Yeah, I have a question for Toren. So I think, aging field, the fasting, intermittent fasting versus caloric restriction is always, a lot of discussion there. So I really want to hear your idea.
Well, I haven't had lunch today, so I actually do the intermediate fasting thing. So I think, AtLab doesn't really do that, but I know Rafa De Cabo's group at NIA, when you calorically restrict mice, basically when you provide them food, they're pretty hungry and they eat very quickly, as opposed to if ad libitum mice, they'll eat over the course of the day. And so they've gone back and look at that much more carefully. And if you give mice the normal diet, but time restrict their feeding, so, train them to eat only a few hours, you get most, but not all the benefits of caloric restriction. So I think at least in rodents, there's pretty good evidence that time restricted feeding is probably, sufficient to induce an increase in lifespan. It's not the number of calories, but maybe how quickly you eat them, or, the window of time that you're not eating that may provide benefit. Again, whether that translates to humans, who knows, basically.
Jennifer, do you have a question, or is that just?
Yeah, no, I have a question for Marc. And actually this is for a more general audience question as well. So in your studies, Marc, when you're looking at senescence, the cells are continuously being fed. They never go through any type of I assume you're
Right.
you're feeding continuously, you're replacing the medium.
Right.
So they never go through any type of nutrient depletion, etc. I'm wondering to what extent that's the case for cells in an organism. To what extent are the nutrients that are bathing cells in different tissues, etc., constant or undergoing fluctuations over time? Through a circadian cycle, through a feeding fasting cycle. My assumption is there's changes inside the organism. What you're looking at, Marc, is where there's no modulation of the nutrients that are available to those cells, which is interesting, but I'm curious as to whether that's reflecting what's happening inside cells that are within an organism.
Well, I think the whole senescence question, I mean, in a 10-minute presentation, I didn't really have a chance to talk about this, but I mean, the whole senescence question is a very open question. The senescent cells are generally, they're not the majority of the cells. There are some cells that are senescent, and they have certain characteristics, and one of the characteristics is they secrete lots of growth factors and things, and so they have effects on other cells, which is some people think may induce their senescence. Other feel that that's part of the aging aspect of the whole organism. So it's a really murky subject. In this case, the phenomenon is that is really a cell culture phenomenon. That's a clearer phenomena where those cells actually also are not synchronously going into that state, but they are going into a state that people analogize with what they see in whole tissues. So I not only can't I answer your question really, except to say that we haven't done anything extraordinary except to produce what's called DNA damage-induced senescence or damage-induced senescence, where cells stop dividing. That's one characteristic of senescent cells, and they keep growing to some degree. And some people feel, I mean, like Angelica Allman, who made a case with yeast, and I try to extend that to mammalian cells, arguing that the swelling of the cell, that is the loss of mass density, was itself the phenomena that was due to damage in replication and was also causing all these secondary effects on other cells. And I think we need to characterize this process better. There's something really very interesting there. There are experiments that have been done that have shown that if you eliminate senescent cells in animals, you can extend lifespan or health span. So all those things are interesting, but and I think the measurement of cell size is also generally not very reliable. And so I think I put this in to demonstrate the technology, but not to stand on any of these experiments to say we really understand this is how fundamental this is to aging. I think it's clearly involved in aging. It's related to aging. It's probably cell autonomous, non-autonomous, but it needs to be really investigated much more thoroughly than it has been.
Thank you.
Ralph?
I had a question about ramen, maybe for both Mung and Marc. Does an association between a nucleic acid and a protein change the properties of either the nucleic acid or the protein detectable by ramen? I guess what I'm wondering is whether this could be a way to monitor ribosome abundance non-invasively. Generally speaking, I mean, measurably, no.
measurably denatured proteins, native proteins, RNA protein complexes, all these things really can be, the Raman signal may be slightly influenced in its, it's very slightly influenced by those conditions, but it's pretty much almost a measurement of a single bond of carbon and hydrogen, if that's what it's tuned for,
Yeah.
and rather insensitive to surrounding effects.
Yeah, so for nucleotide, I think you're particularly, that's why I asked about fingerprint region for CH, because in CH, you don't get much signal from nucleotide. But if you go to fingerprint region, you can get much richer signals there. You even possible to distinguish like ATP versus ADP or AMP. But most of, like right now, the technology challenge is when we go to the fingerprint region, the noise is very high. So then, the imaging is more challenging than the CH region. So in terms, the neighboring environment, how that affect the chemical bond. So at least you have another chemical bond next to that chemical bond. Otherwise, binding to a protein or not, if you don't change the chemical bond, you won't cause changes in the signal. Yeah.
Thank you.
I should point out one thing which was Sunny G adopted, which made Raman microscopy possible, was this idea of pumping the energy into the system with a separate laser band, and it's called stimulated Raman microscopy, and that increased the sensitivity 100,000-fold.
Yeah.
And that is what really enabled you to go from analytical chemical method with a path length of a centimeter through a cuvette to actually making measurements in cells.
Yeah. So before, like if you do spontaneous Raman for one 512 by 512, you maybe need like 10 or 20 minutes. Now with the stimulated Raman, you only need 1 second.
Less than 1 second.
Depending on how fast the microscope scans. So I think the limitation becomes
much less than 1 second.
Christine? Oh, you're muted.
Yeah, ID was first.
Ah, okay. In that case, Heidi.
Yeah, well, Christine can go first, doesn't matter. I have a question for Karin and probably Ralph at the same time. So when you look at mitochondrial disorders, we've seen that like a lot of different mitochondrial disease fibroblasts start to give us weird senescence phenotypes and this and that and the other thing. So is it known in beta oxidation disorders that there's a senescence issue, or has that been observed before in monogenic disorders of beta oxidation, or is that how would that be seen?
Yeah, I don't think anyone has studied this very closely with respect to beta oxidation. To say, most of the beta oxidation disorders are relatively well tolerated if you avoid fasting periods, with the exception of some of the diseases that cause accumulation of intermediates that may be toxic, with the 3-hydroxy acylcarnitines being probably the best example. I think Eric Schoen, though, had data years ago suggesting that maybe there was some senescence in cells in the brain that had OXPHOS defects.
Yeah.
Maybe someone remembers that better than me. This has gone back quite a few years now.
Most of the CPT disorders affect the heart, in patients, and that's a post-mitotic organism, so it's hard to see senescence basically there. So it may be, that the tissues that are most affected are non-dividing basically.
Yeah.
So, and how do you it's hard to, the people have tried to define senescence in cardiac myocytes, but it's actually not clear that it's hard to get a definition in vivo of what senescence is, and it's particularly hard when the cell is not dividing under normal conditions.
Yeah, it's just because of some of the fibroblast lines, if you try to challenge them with starvation, because often these mitochondrial disease lines will be fine, and then you challenge them and crazy stuff happens. And so it's making me rethink a lot of stuff. I have one quick question also for Toren. So when you were, and maybe you've published, I'm sure you know this already, but what kind of proteomics changes? This is coming back to Jared's question of yesterday. When you start to manipulate amino acids like methionine and all these kinds of things, can you identify SLC transporters that change? Does the mitochondrial proteome change selectively? Do you have any insight into that?
When in methionine restriction?
Or any of your sort of amino acid experiments where you're selectively altering?
Yeah, I mean, we did RNA-seq. I can't remember if any of the SL transported proteins were predominant. I'd have to go back. I'm sorry, I just don't remember if that was the predominant RNA transcript. But it doesn't ring a bell, but it could have been there. I don't know.
Just could be a model to try to identify some selective protein turnover mechanisms to adapt to those things. But anyway, thanks. That's all.
Okay, Christine, now you.
Yeah, my turn. Okay, well, it's a bit in the same line as ID because it came to my mind that when Karin said that CPT1 inhibition could rescue viability in their cell, I think it may be that trimetazidine versus, let's say, inhibition of CPT1 may affect just the acylcarnitine level. And this reminds me of a paper from a colleague of mine, Bruno Lefort and François Labarthe in Tours in France, where they could see that when they inhibit carnitine palmitoylase transferase in fibroblasts from patients with mitochondrial trifunctional protein deficiency, they can rescue mitochondrial oxidative phosphorylation. So I think one thing is we are talking a lot about fatty acid oxidation, but it's also the efficiency of this process, whether you will see these acylcarnitine long chain accumulation in cell. And if you inhibit CPT1, you would prevent this because it comes with the CPT2 activity. So I think maybe from your metabolomics, you may not have seen the acylcarnitine because it depends which method you chose, but maybe it's something you could look at.
Yes, indeed. So we actually chose on purpose to look with a more polar metabolomics method, a classical one, because we wanted to be more open to also finding non-lipid related changes. And there it was the more obvious choice to go for a non-targeted method. We do capture some mostly polar lipids, but we measured in negative mode, and so positive mode measurements would have been more suited. What we actually see is a lot more phospholipids that have these long chain fatty acids incorporated. So we think in part those are actually being redirected. So I think there's many different things going on. As was mentioned yesterday, metabolism has many different ways of compensating for what we do to it in a way.
And also, depending which fatty acid you have in your medium, let's see if you would have oleate versus palmitate. We know that they have differential effect on ceramide synthesis, and this can also influence the viability. So thank you.
Toren, if I can ask you one question, you at the very end of your talk, your slides were quite sobering about the differences between humans and other model systems with respect to caloric restriction. And so where's your nickel in terms of what the difference is between humans and these other organisms?
I think it probably has, I think very simply it has, I think going back to, the magnitude, if you starve a mouse, the magnitude of effect on body mass is just tremendous. Obviously losing 20% of your body weight overnight. Like I say, we know there would be no bariatric surgery. If you could, as a 200-pound person, if you could lose 30 pounds a day, just from, not eating that day. I think that's the scale of the physiology is just so different, and I think, the evolutionary arguments also made, about, in animals that, have a very short reproductive window of a year or two, that reproduce only at certain times of the year, that caloric restriction, evolutionarily might have a much bigger advantage. Animals that could adapt to caloric restriction, they might have, anyway, I think there's, body surface area, reproductive length, all those sort of things, I think make our physiology so much different. And I know we had this discussion yesterday about model organisms, but, and what organisms might be the best for genelia, but, there's really no substitute for, as Ralph sort of pointed out really nicely, there's really no substitute for human physiology. And, everything else, you sacrifice every time you get away from the humans, basically, in terms of insight.
Sure. So we should probably close this final session now. We're a few minutes over, but I think that's okay. And this also brings us to the end of the entire two-day workshop. And I think I speak for both Jennifer as well as for Rong when I say that we've really enjoyed putting this together. And we just really want to thank all of you, all 24 of you that gave terrific talks over the last 2 days. I know that this was a big ask, especially because this is August, and also because these are a little bit unusual talks that really asked you to sort of push yourself and think about the future. So we really appreciate everyone's time and effort, all the active participation, and I think it's been a great 2 days. And hopefully a lot of these conversations are simply going to continue in the days, months, and years to come. And finally, I want to really thank Janine for all of her help. None of this would have been possible over the last few months, weeks, or over the last 2 days. So thank you, Janine, as well as HHMI, for providing us with this forum to explore the future of metabolism and nutrition. So I hope everyone has a great summer, and I'm going to hand it over to Janine now.
Great. Thanks so much, Vamsi. On behalf of Janelia, we thank you and Jennifer and Rong for putting together such a great workshop, and of course, all of the speakers and discussion leaders. And we also thank the audience for joining us these past 2 days for these really inspiring sessions. So for all of you who may be interested, you can tune into our website at 4dcpjanelia.org if you want to receive updates about our 4D cellular physiology program. If you want to see the workshops that we have coming up, please check in there. And also the recordings of the talks and discussions from this workshop will be available on Janelia's YouTube channel within the next couple of days. So check it out. Thanks, everyone. Panelists, don't go too far. We're going to take just a quick break, and we will start our closed panel discussion at about 3:15 p.m. Eastern. And I just sent an email with the link. So we'll see you then.